I'm Brinda Vallat. I'm a scientific software developer at RCSB Protein Data Bank. And today I'm going to be talking about finding, accessing, and analyzing computer structure models from rcsb.org. All right. I have a brief outline here, but I realized Stephen already introduced what computer structure models are, so I will kind of skip through that quickly and move on to explaining the models that are currently available in rcsb.org, and then show a live demo to illustrate the tools and functionalities that we have on rcsb.org. And I will focus on case studies that have already been introduced by Chris, Janaka, and Dennis. The disease-resistant RPP13-like protein from Arabidopsis, the riboflavin biosynthesis pathway from Arabidopsis, and the Micrococcus luteus aldehyde dehydrogenase. What are computed structure models? We already saw part of this slide in Stephen's talk. These are, so I won't spend too much time, just to quickly catch up. These are predicted models of macromolecules obtained using computational methods. And the two recent methods, AlphaFold2 and RoseTTAFold2, are ab initio prediction methods. AlphaFold2 is developed by Google DeepMind, and RoseTTAFold developed by Howard Hughes medical investigator David Baker and his team at University of Washington. These methods achieve accuracy comparable to low-resolution experimental structures, and their success was enabled by recent breakthroughs in applying artificial intelligence and machine learning approaches to structure prediction. These methods have been trained using thousands of sequences and structures of known proteins from the PDB. So the primary data for training these methods is coming from the PDB. So without the enormous wealth of curated and validated 3D structure data from the PDB, the application of machine learning techniques to train these systems would not have been possible. AlphaFold protein structure database provides open access to AlphaFold2 predictions. The database is hosted at the European Bioinformatics Institute and includes over 200 million protein structure predictions, providing a broad structural coverage of sequences in UniProt. The Model Archive is a repository for computer structure models referenced in publications and is hosted at the Swiss Institute of Bioinformatics and includes predictions of core eukaryotic protein complexes modeled using a combination of RoseTTAFold and AlphaFold generated by David Baker and his team at the University of Washington. So these two are the primary resources for the computer structure models, which have now been integrated into rcsb.org. I want to quickly touch upon model quality metrics when it comes to computer structure models. Although these computer structure models generated with the recent machine learning approaches have become increasingly accurate, it is important to look at the model quality estimates for these predictions, because these can be the estimates can be used to evaluate these models and assess their suitability for specific downstream applications. Computer structure models in AlphaFold and Model Archive include two types of model quality metrics. The first one is a per residue confidence measure called the Predicted Local Distance Difference Test, PLDDT, provided on a scale of 0 to 100, 100 being the best. As can be seen here, regions of high and low confidence based on PLDDT are identified using color codes. The blue and cyan regions are the regions of high confidence, and the orange and yellow regions are regions of low confidence. The PLDDT score can be averaged over all residues in a protein chain to provide a global score. The second metric is the predicted aligned error, or the PAE score, which is the expected position error at residue X when the predicted and true structures are aligned on residue Y. PAE enables the assessment of residue contacts as well as domain orientations. So these are the two kinds of quality assessment metrics that are available from AlphaFold and Model Archive. Before delving into the computer structure models available in RCSB.org, I want to give a brief introduction to the model SIF data standard. Dennis mentioned the PDBX MMSIF data standard used by the PDB archive. Model SIF is an extension of PDBX MMSIF for computer structure models. Definitions for representing small molecules and macromolecules and atomic coordinates are all taken from PDBX MMSIF. And definitions pertaining specifically to computer structure models, including definitions of targets, templates, alignments, coevolution data, model quality metrics, all these are defined in Model SIF. Model SIF is publicly available via GitHub and is managed by the Worldwide Protein Data Bank Model SIF Working Group. Importantly, the Model SIF data standard is supported by both AlphaFold2 and Model Archive. Moving on to computer structure models in RCSB.org. This summer, we released an updated RCSB.org research-focused web portal, which now delivers over 1 million computer structure models, together with the approximately 200,000 experimental structures already available in the PDB, thus creating a one-stop shop for studying macromolecular 3D structure data. This was made possible because a Model SIF, which is an extension of PDBX MMSIF, facilitates interoperation with PDB data. The extensive set of tools and services provided by RCSB.org can be used to search, analyze, and visualize computer structure models alongside experimentally determined PDB structures. These computer structure models in RCSB.org are consistently distinguished from the experimental structures through source-specific icons and unique coloring schemes. Experimental structures are identified using the blue Erlenmeyer flask icons, as you can see here, and the computer structure models are identified using the cyan computer monitor icons shown here. Provenance information about these models are clearly provided, and we have extensive documentation that explains how to access and analyze these models alongside the experimental structures. So what are the models that are currently available that have been integrated with RCSB.org? The current set consists of a prepackaged collection of about 1 million models from AlphaFold2, as well as 1,100 models from Model Archive, comprising of the core eukaryotic protein complexes modeled using RoseTTAFold and AlphaFold2. These computer structure models collectively cover the entire human proteome and those of model organisms, selected pathogens, as well as organisms relevant to bioenergy research. I will now move on to the tutorial, the live demo part of this session, and showcase a few case studies that have been introduced by Dennis, Janaka, and Chris earlier today, including the Arabidopsis thaliana resistosome, the riboflavin biosynthesis pathway, and the aldehyde dehydrogenase from Micrococcus luteus. My goal here is to use these case studies to demonstrate the tools and functionalities that we have on RCSB.org to study these computer structure models. So now I will be moving on to my browser. Okay. So let's start with the first example, the disease resistance protein from Arabidopsis thaliana. I just want to copy the term that we want to search. We start with the top bar search. The first thing that I want to point out is including computer structure models is an opt-in button here. You got to turn that on if that needs to be included in the search. We'll do that right now. And then I'm going to paste the search term. Auto suggestions come up here, and it's always recommended to use these if they are relevant to your search. So let's start with the uniprot molecule name for this specific protein. And this launches the search query that returns 29 structures. A quick look at the refinements panel shows that there are 24 computational and 5 experimental structures. The refinement panel has terms that are, now that we've included computer structure models, there are some specific attributes that are relevant to computer structure models. You can filter based on the source database or the global quality metric PLDDT score. Or you could look at just the computational or experimental structures. In addition, the search results can be sorted based on either the global quality metric, so then you can see the best scoring ones on top, or you can sort based on, okay, you have a combination of experimental and computational structures, but say I want to look at experimental structures first, you can do that, and the results would be sorted accordingly. As I mentioned earlier, the experimental structures and computed models are distinguished clearly using color schemes and icons. You can see that here. And if you scroll back up, you can see that the advanced search query is populated with the query term that we actually started the search with. And let's say we want to now look specifically at Arabidopsis, we can filter for Arabidopsis. And now there are only 6 structures. This includes the 5 from experimental and 1 computational structure, computational model. The advanced search query builder also has specific attributes for computer structure models. You could search based on the source database, global quality scores, or identifiers from the source database, either from AlphaFold or AlphaFold.db or Model Archive. All these can be used to build into the query builder, and complex queries can be built using other scientific search queries like sequence similarity or structure similarity, all these that are useful even for experimental or if you're searching for experimental structures as well, like Dennis already pointed out. Now let's look at a computer structure model, the structure summary page, complementary to the experimental structure summary page. It has a little less information than experimental structures that Dennis demonstrated a little earlier, but I would like to point out a few specific attributes that are highlighted here. One is the model confidence, the global confidence score, and the distribution of local confidence score based on the color scheme shown here. The image also follows the same confidence PLDDT-based confidence color scheme. And you have a similar macromolecule section to what you see for experimental structures, that you can launch different kinds of queries from this page, and also the 1D protein feature view. All right. Now, I would like to move on to the next case study, which is the Micrococcus luteus aldehyde dehydrogenase. So in this case, I want to start with the sequence. So I'm going to the advanced search query builder. Sorry, I want to launch the sequence similarity search. I have the sequence of this protein. Let's use all the default parameters, but I want to return polymeric entities, and I want to include computer structure models. And if you launch that search, that retrieves 666 entities here. But what's of interest is that the highest scoring one has a sequence identity of 48%, which means there is no 100% match. So this exact sequence, there is no structure currently available either in the experimental PDB archive or the computer structure models that have been integrated. So at this point, you could either use one of these experimental structures as templates to build a homology model, or you could use AlphaFold software to build an ab initio model. So using the KBase pipeline, we have an AlphaFold model built for this structure, and the next the interesting question here now is, okay, I have a model. What else can I do? The sequence search has given me a lot of interesting results. Let's say we pick an example here, and this happens to be the structure of the bifunctional enzyme from E. coli. And this, this structure has small molecules bound, crotonal coenzyme A and NADP. So what we can do is, okay, this turned out to be structurally, I mean, in the sequence search, this is a close homolog. So we could do a pairwise alignment. We have a pairwise alignment tool on rcsb.org. So let's launch that. And okay, so the E. coli enzyme is 6JQO. So the tool allows you to either choose an entry that's already in the database or upload a structure that you might have. So let's choose this one, 6JQO. And I have I'm going to choose upload file here. And I have the Micrococcus luteus aldehyde dehydrogenase that was modeled using AlphaFold, that was shared with me by QZ. And the pairwise alignment tool has several options for the alignment, but for now, for this demo, let's just choose the default option and hit compare. This is going to, this takes a little bit of time, so, okay. So we have the E. coli enzyme and the uploaded file that has the M. luteus AlphaFold aldehyde dehydrogenase model. And if you scroll down, our MolStar visualization tool has now shows the aligned structures, and you can visually see that they are very similar. In addition, you could click on the scores tab and you can see that the RMSD is 1.31. There's a TM score 0.93, which is very significant. So these are structurally similar. By default, the view is, it shows the aligned residues. But if you use the pull down menu here and look at structures, what it does is the E. coli enzyme is now superposed, that the entire assembly is superposed to the Micrococcus luteus AlphaFold model. And now there are ligands, small molecules present in the E. coli enzyme. We already saw that. So now what you could do is you can focus on these ligands using the MolStar tool. Shuchi will explain in more detail about how to use the tool, but I just want to quickly highlight how you can get some structural and functional insights using the tools in rcsb.org. So now you can focus on the ligands, and so there are ligands in the aligned regions here, the crotonal coenzyme A and NADP. So this you can focus and you can get some additional insights into the ligand binding and other functional insights into the aldehyde dehydrogenase and what kind of ligands can bind. So this kind of information that experimental structures have these small molecules bound and the computational models don't have that. So doing this kind of a pairwise alignment can give additional insights into small molecule binding. All right, so the next story that I want to that I want to point out is the pyridine binding, the pyridine degradation pathway for the same enzyme. We want to see if we can find structures that have pyridine bound. So let's start with pyridine again. I'm going to use the auto suggest. Scroll down to in chemical name pyridine. Okay, let's turn CSM on. So that pulls up several structures, but it could be a derivative of pyridine as well. We want specifically pyridine, and that happens to be this ligand 0py. Let me just show you the ligand page. This is the ligand page for pyridine, and it says it's present as a standalone ligand in 3 entries. And these are the 3 entries that have pyridine. So for example, let's click on the first one and go to the small molecule section. There's pyridine here. A cool feature here is if you click on this ligand interaction button here, that takes you to the 3D view directly, but it focuses on the ligand and the interacting residues. So you can already see the pyridine ligand and the residues in the protein that are interacting. And what we can do from here is we could do like a structure motif search. We can launch a structure motif search, but what that means is you can select some of the residues that are interacting with the ligand. Say, for example, I can choose some of these residues. As I'm choosing, you can see that the query, you can see what is being chosen here, and then you can launch a search right from this, the Molstar viewer here. I already did this search, so I want to, yeah, okay. So I had picked randomly a few residues from the ligand binding motif, and then launched the structure motif search. And that brings up 481 results, and all of these have this motif, but in this case, I just randomly picked a few. You could look at closely which are the residues that are interacting with pyridine and search for that particular motif. And this now has a included computed model. So now you can see there are computed structures as well as experimental structures that have this motif, and you can look at the results in detail and see which of these computational structures that have this pyridine motif, and if there's any overlap with the sequence search that we started with, to compare the results of the sequence search with this struct motif search to see if any of the structures of interest have this particular motif. So this also can give additional functional insights into ligand binding in computed structure models. Okay, so the last story I want to focus on, let's go back to, is the riboflavin biosynthesis pathway. So we saw that we have the E. coli enzyme, which was, I think, from Chris's story, it was 207P. Let's make sure that's what it is. Let's see, 207P. Okay, so that's the E. coli enzyme. So I want to, and then we have the 207P chain A, and now we have the Arabidopsis enzymes, the deaminase, and the reductase. So I want to pick one. I have the identifiers here, so I just want to, okay, chain A, and then I can launch a pairwise alignment again. Okay, so this is the E. coli RibD with the Arabidopsis, the deaminase AlphaFold model. So now we can do, again, a structure comparison, pairwise alignment, to see how these structures match. The scores look good, and then you do the same thing, look at structures so that you can highlight the ligands and see where the ligands can bind in the computed model. You could do the same thing with the reductase. Let me go and get the accession code for that. All right. Right. And then I could do a comparison. Okay, so that's the E. coli RibD with the Arabidopsis reductase. This one is not as good a match as the deaminase, but still it's reasonable. A TM score higher than 0.5 essentially means a similar fold. And you look at the aligned residues. Yep, looks reasonable. And then you can look at the entire structure. And now you have the context to look at ligand binding and delve into the details of, okay, where can ligands bind, small molecules bind, and what kind of functional insights you can get. Okay. So one other thing that I wanted to point out before closing is you can also do a structure similarity search. This is another tool that you can launch from an entry page. So if you click on that, and I always recommend to go and look at the advanced search query builder when you're clicking on any of these query by example to know what search is being carried out, and you can always build on top of this, add subqueries. And let's turn this on and search. So in this case, it's doing a shape search or a structure similarity search and coming up with both experimental and computational structures that share structure similarity to our query here. In this case, I think I ended up using the Arabidopsis bifunctional, no, not the, sorry, the E. coli bifunctional enzyme. And based on the results that you get, again, there is a structure match 3D view button here. And when you click that, it'll take you directly to the pairwise alignment and align the query to the result that we picked. And you can see the structure similarity again. All right, so we looked at different tools, the pairwise alignment tool, the motif search, sequence search, structure similarity search, the different kinds of scientific searches that you can carry out using the RCSB.org web portal. And this brings up, I mean, you can include computer structure models or focus on experimental structures depending upon the scientific question. So to quickly summarize, RCSB.org now provides access to over 1 million computer structure models from AlphaFold2 and RoseTTAFold alongside over approximately 200,000 experimental structures of macromolecules. Various search, analysis, and visualization tools available on RCSB.org facilitate the study of these models together with the experimental structures to obtain additional structure and functional insights into the binding sites, etc. And depending upon the scientific question being investigated and the data available to address this question, for example, sometimes you might only have the sequence, or sometimes you might have already an experimental structure, or sometimes you only have you have good templates from which you can build a model. So based on the question that's being investigated and the data that's available to address the question, different search and analysis strategies can be devised using the tools available on RCSB.org.